Okay, so welcome back to this next video in which we are discussing Huntington's disease and complexins. Right, okay, so we've discussed these two complexin knockout mice. Complexin 1 knockouts have this very severe phenotype where they get ataxia and seizures, whereas complexin 2 knockout mice have this much more subtle cognitive defect where they're not capable of relearning an experimental setup like this. They're not capable of learning that the rules that they originally learned have changed and that they need to change their understanding of those rules. Okay, so finally we've missed off one type of knockout, which is if you knock out both of them. So if you do a double knockout, if you make a mouse that has no complexin 1 or no complexin 2, well, I've already told you that this is fatal. So basically the mice die when they're at birth. Okay, so they survive in the uterus, but when they are after birth, they die. And the reason they die is that they don't breathe. And the reason they don't breathe is that if complexin 1 and complexin 2 are both gone, then the neurons in the phrenic nerve, which innervates the diaphragm, those don't release enough neurotransmitter onto the diaphragm to cause it to contract properly, so you don't breathe, basically. So they die because they don't breathe. Okay, so double knockout is the most severe. Right, okay, so now what I want to tell you about is how this complexin 2 knockout here actually has very similar phenotypes to our mouse model for Huntington's disease. Okay, so let me tell you about that. Right, so the mouse model for Huntington's disease is a type of mouse known as an R6/2 mouse. Okay, so that's the inspired name for a mouse that has Huntington's disease, an R6/2 mouse. Okay, right, so let me explain to you a little bit about Huntington's disease, and it will be a little bit because there's very little known about Huntington's disease. We know what the phenotype is because we have people suffering from it. We don't know much about the cause of it. Okay, so Huntington's disease then, it's caused by well, actually I think I should write out the name. So this is Huntington's disease. This is our mouse model for Huntington's disease. Huntington's disease. And Huntington's disease was once called Huntington's chorea. So if you hear people refer to it as Huntington's chorea, that's the same thing. Chorea basically refers to involuntary dancing movements. So if someone is dancing effectively, they do these very sort of elegant, smooth movements that looks like dancing, but they have absolutely no control over it. That's what chorea means, and that chorea is one of the phenotypes of Huntington's disease in humans. Okay, so basically, in Huntington's disease, it results because of a mutation, a problem with the Huntington protein. Well, not necessarily a mutation, it could have been inherited, and usually it is inherited, but there's a problem basically with a gene that codes for a protein known as Huntingtin. Okay, so this gene for this protein, so I'll write the name of the protein first. So the name of the protein is Huntingtin. Okay, and this is often abbreviated to HTT, like that, Huntingtin. Right, and the gene for this protein, Huntingtin, is on chromosome 4. Okay, so let's draw our chromosome 4 here. So chromosomes, remember, have these long and short arms. So they have a short arm and then a long arm. And the short arm is called the P arm for petit in French, petit, which means small in French, and then Q is the large arm here. Okay, and the Huntingtin protein is on the short arm of chromosome 4. So somewhere on chromosome 4's short arm will be the gene which codes for this Huntingtin protein. So here it is. This is the Huntingtin protein. So, oh well, the Huntington gene rather. HTT gene. Okay, right. So now let's draw it out in a bit more detail. So let me draw my drawing of DNA here. Okay, and let's say this portion represents the Huntington gene. So this is the Huntington gene. Okay, here that I've drawn here, and it would be longer than that in reality. But to save paper, I'll draw it like that. So this is the Huntington gene. Right, so what actually happens in Huntington's disease? Well, basically, if we look now at the actual organic base sequence of the Huntington gene, then there will be a huge bit at the front here that I don't care about. But what happens towards the end of this Huntington gene is that you start getting just a repeat. You get CAG, and then you repeat it over and over again, CAG, and you continue repeating it. So towards the end of the Huntington gene, you have this, what's known as a trinucleotide repeat. So you repeat this CAG many times, and therefore it's called a trinucleotide because you've got 3 nucleotides, and then it's a repeat. So a trinucleotide repeat. Okay, now when you come to actually translate this gene, i.e. firstly you'll make an mRNA copy of it. So I've only drawn one strand of the DNA here. Of course there will be another complementary strand here. When you come to make an RNA copy of it, you will copy this out, and then when you transcribe that, this CAG codon, as it is, so here's our piece of mRNA, okay? So basically, when you make the mRNA, you use this other complementary strand as your coding strand. So this blue strand is going to be your coding strand, i.e. you will make the mRNA complementary to the coding strand. So the RNA polymerase will work its way along the coding strand and will build a piece of mRNA that's complementary to the coding strand, i.e. the same as this other strand that I originally drew, which is in orange. So basically, you'll make a piece of mRNA which has the same sequence of organic bases as this strand of DNA here, the non-coding strand. So this is the non-coding strand. Okay, but it's the strand which actually has the correct sequence of organic bases for the piece of mRNA. Factor in the fact that you have to turn thymine organic bases to uracil. So here's our mRNA. And when you actually translate this piece of mRNA, i.e. you put it through a ribosome and the ribosome turns it into protein, this codon here is going to be read, and it codes basically for, well, it codes, another tRNA is going to come in. So if this is the ribosome coming here, what's going to happen is you're going to get a tRNA coming in with the complementary anticodon, which will be GTC here, okay? And the amino acid that will be attached to that tRNA is going to be a glutamine, which has the single letter code Q. So this is going to be a glutamine. So basically, this repeat of CAG, 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 this trinucleotide repeat at the end of the gene for Huntington's, is going to code for absolutely loads of glutamine amino acids on the end of the protein. And basically, when we make the proteins, let's get our protein here. So here's our protein. And initially, I'll just draw it as like a line. So this is our polypeptide down here. So this is the initial bit of the polypeptide in blue, which I haven't gone into detail of. And then we're going to have just these glutamines in a whole tail like this. You're going to have glutamine after glutamine after glutamine, which I'm actually drawing as little blobs. Okay, so these are all glutamine amino acids joined together at the end of this polypeptide. Okay, and this tail of glutamines is what's known as the polyglutamine tract. So this is the polyglutamine tract. Glutamine tract. And because glutamine's single letter code is Q, this is often known as the poly-Q tract. Okay. So on the end of a polypeptide, you will have, well, on the end of the Huntington polypeptide, HTT, you will have this polyglutamine tract. Now, if the number of times this trinucleotide repeat is repeated is less than around 26, then you are normal, okay? So less than 26 repeats of CAG is normal, and you'll have 26 you'll have 26 or less than 26 glutamines on the end of your Huntington protein. So you'll have a nice short polyglutamine tail, and your Huntington protein will perform its normal function, and you'll be perfectly happy. Okay. Unfortunately, if you have greater than 36, and I mean that, greater than 36, don't think I've somehow got confused here. If you're in between, if you're between 26 and 36, then you're in the intermediate zone where it's sort of foggy as to whether you'll get the disease or not. But if you've got greater than 36 of these trinucleotide repeats, i.e. you've got greater than 36 of these glutamines on the end of this Huntington protein, then you will get Huntington's disease. By about the time that you're 40, you will have developed symptoms of Huntington's disease. Okay, so basically this is why these online companies which offer to sequence your exome generally for you, and you can have it done for, I think now, about 150 quid. You can have your entire exome sequenced. These companies at the moment are completely unmoderated at all, and they are giving people information that could be very, very distressing for them, because they could tell them whether or not they're going to get Huntington's disease in later life. I'm 20. I do not want to know whether I'm going to get Huntington's disease when I'm 40. But I could find out. I could go onto these online websites, and they are genuine. These people are genuinely sequencing your DNA. All you need to do is spit into a tube, and they will be able to tell you whether you're going to get Huntington's disease. And this at the moment is completely unmodulated. You're not providing these people with genetic counselling afterwards, and you're potentially telling them extremely distressing information. Okay, so that's a little bit aside on the ethics of genetics, but this basically, if you get too many glutamines on the tail of your Huntington protein, it causes the function of this Huntington protein to completely change. So the whole function of the Huntington protein changes if you have greater than 36 glutamine residues in your polyglutamine tail. And now, instead of being called Huntington, what it's instead called is mutant Huntington. So this Huntington protein, which isn't actually functioning in the normal physiological way, but is instead doing loads of other bad things, is instead referred to as mutant Huntington. And this is Huntington with over 36 glutamines in this polyglutamine tract. So this is MHTT for short. Right, so this mutant Huntington is going to cause the symptoms of Huntington's disease. Now, what are the symptoms of Huntington's disease? Well, basically, it's neurodegeneration. You get neurodegeneration all over the brain. Neurodegeneration. It's not limited to just one area of the brain. All of your neurons all over the brain start to die, basically. They start to degenerate. Okay, there are certain areas of the brain that are prone to it, that degenerate first, specifically the basal ganglia, and we'll look at the function of the basal ganglia and why their degeneration leads to chorea later. But in actual fact, the entire brain degenerates in Huntington's disease gradually. It's just that certain areas degenerate faster. Okay, so why? Well, there are many plausible explanations, but we're going to look at the explanation involving complexin 2, because remember I told you that these R6/2 mice, which have this mutant Huntington, so these are mice with mutant Huntington, the disease phenotype that these mice get is exactly the same as the disease phenotype that these complexin 2 knockout mice get. I.e., when you do this two-choice swim tank experiment, the the mutant Huntington mice, these R6/2 mice, have exactly the same problem, i.e. they cannot relearn when the rules have changed. Okay, right, so what we then did is we looked into this. We looked into, does mutant Huntington cause some sort of problem with complexin 2? And it does. If you have mutant Huntington, what we have found is that it stops the function of complexin 2. So basically it inhibits complexin 2 function. Okay, so we found that in mice with mutant Huntington, these R6/2 mice, they do have lower levels of complexin 2 in their neurons, and it's because this Huntington, this mutant Huntington, is somehow leading to its production being reduced. Now, what would you then predict? If the theory we've now come up with then is that in Huntington's disease, in these R6/2 mice, they have this mutant Huntington. The mutant Huntington stops the function of the complexin 2, which then leads to reduced exocytosis, which then leads to neurodegeneration. And indeed, Huntington disease mice do have reduced exocytosis. You see that these R6/2 mice, they do have impaired exocytosis in their neurons because of the lack of complexin 2. Okay, but we would now expect, basically, if you returned complexin 2 into their neurons, then exocytosis would be restored. Okay, so if you actually do that, if you isolate neurons from these mice which have Huntington's disease, and you restore complexin 2 into them, then their exocytosis returns completely to normal, basically. So we have found that in Huntington's disease, in mice with this Huntington protein that is mutant, you have reduced exocytosis in all of your neurons, and it seems to be because complexin 2 is lower. Firstly, we have measured complexin 2 and found that it is lower, and secondly, we have found that that if we return complexin 2 back into those axons which have the mutant Huntington, it restores the exocytosis. Whether it's the drop in the complexin 2 that actually leads to the neurodegeneration and the death of the neurons over time, we don't know. But potentially it could be. So complexin 2 seems to be at least one of the targets of this mutant Huntington, and it seems to result in reduced exocytosis in the neurons within these mutant Huntington mice, these R6/2 mice. Okay, so in the next video what I want to do is just give you a little bit of insight into the basal ganglia and how they function, and how neurodegeneration in those regions of the brain, which happens first in people with Huntington's disease, leads to chorea, these involuntary dancing movements.